Welcome everyone. Today's topic is bacterial cell structure, external structures and types of cell walls involved in this. So first of all, how are prokaryotes different from eukaryotes? So the main difference, the prokaryotes means where the nucleus is not covered with an envelope, whereas in case of eukaryotes, they are having true nucleus, which means nucleus is covered or present in an envelope, that is nuclear membrane. And in case of bacteria, we will see the cell wall is made up of peptidoglycan, and whereas in archaebacteria, that is it is made up of polysaccharides, and in case of eukaryotes like fungi, it is made up of chitin, etc. So mostly in case of prokaryotes, there is no true nucleus, and also they are not wrapped around with histones. So we'll see what are the external structures present in case of bacteria, that is prokaryotic cell, and cell wall, what kind of cell wall it is, and we'll see what are mainly in this video, what are the external structures and their functions, and cell wall type, and what are different types of cell walls in case of bacteria. So structures common to all bacterial cells, that is cell membrane, cytoplasm, ribosomes, that is 70S type ribosomes, and chromosome. Structures found in most bacterial cells, that is cell wall and surface coating or glycocalyx or caps, which includes capsule or slime layer. So glycocalyx is of two types, that is capsule and slime layer. Coming slides we'll see what are. So structures found in external structures found in bacterial cells. Those are flagella, pili, fimbriae, capsules, slime layers, inclusions, actin cytoskeleton, and endospores. So here you can see the detailed functions of each component of bacterial cell. You see the first one, glycocalyx, which is a coating or layer of molecules external to the cell wall. So glycocalyx is of two types, as I already said, that is slime layer or capsule. In some bacteria, glycocalyx is loosely arranged, which is called a slime layer, and in some bacteria, the polysaccharides, this glycocalyx is made up of the polysaccharides, and this is very compact in case of capsule forming bacteria. Next structure is internal structure, bacterial chromosome, and it is called nucleoid. We won't call it nucleus because as the genetic material is loosely spread across the cell, it is called nucleoid, DNA. Next one, plasmid, which is extra chromosomal DNA, double stranded DNA, which is extra chromosomal in nature. It is called plasmid. Next, pilus, and which is an hollow appendage, which is used in the conjugation process for the transfer of DNA from one bacteria to another bacteria. So coming to the ribosomes, here we already said, I already mentioned it is they are having 70S types of ribosomes. And actin cytoskeleton, these are the long fibers of proteins that encircle the cells just inside the cell membrane, and these are responsible for the shape of the cell. Next one, flagellum, these are the specialized appendages attached to the cell by a basal body that holds a long rotating filament. So flagellum contains filament and basal body, etc. In coming slides you will see. The fimbriae are also hair-like bristle structures, and these are also help in adhesion to another structure or other surfaces. Inclusions bodies are nothing but these are the structures where nutrients are stored in case of, under unfavorable conditions, they mostly in favorable during favorable conditions, bacteria store these as a reserved food materials and which can be utilized during unfavorable conditions. Next is cell wall. Cell wall that already will in further slides we will see there are types of cell walls which differs from gram positive and gram negative. And outer membrane, this one we'll see in the further slides. And endospores are these are the resting structures formed by some bacteria where when unfavorable conditions arise, bacteria they form endospores within themselves and which can survive under extreme conditions, and during favorable conditions again they will start germinating. So these are the external structures appendages. Here you can see cell extensions common but not present on all species. There is such example flagella or pilus. Flagella, and there are many types of arrangement of flagella. Here you can see monotrichous flagella, which is single flagellum, lophotrichous, and bunch of flagellum at one end of the bacteria, and peritrichous, where you will see randomly dispersed flagella across the cell. So these are various arrangements of flagella. So flagellum structure, here you can see the flagellum structures. It contains flagellum consists of 4 rings. So 2 rings are present in outer membrane and 2 are present in cell wall in case of gram negative bacteria, whereas in gram positive you will find only 2 rings, and it contains flagellum contains structure like hook, filament, and basal body. So these are the flagellar arrangement, which is through electron microscopy. This was taken 10 images. So here you can see the types of flagellar arrangement: monotrichous, atrichous, amphitrichous, which includes single flagella at both ends, lophotrichous, tuft of flagella at one end, peritrichous, and cephalotrichous. Cephalotrichous means tufts of flagella at both ends. So pili, so as it is made up of protein called pylene, found in gram negative bacteria and used in conjugation process. See the pylene fimbriae structures here. Fimbria, these are the bezel-like fibers. They also contain protein. They are made up of proteins, and they tend to stick each other onto the surface. They are used for attachment. So as I already told, the glycocalyx, which is made up of polysaccharide, they protect the cell from dehydration, and also they protect from bacteriophages, and they also protect the loss of water and nutrients. You can see the slime layer is a loose shield, the capsule is a tightly bound structure around the cell. So you can see the slime layer and capsule, which is a compact structure. So they protect the bacteria from phagocytes, and they are also important in the formation of biofilms, where it is a signal where bacteria forms some communities, and they form a film like biofilms, nothing but the group of bacteria attached. They form like a community where uh, they transfer some messengers. So they are also responsible for signaling in case of bacteria. So basic cell wall, it is made up of 2 or 3 basic layers. First of all, bacteria is made up of cell wall, we know. So commonly it is having cell membrane and outer cell wall. So in gram positive, the cell wall is made up of 2 layers. In case of gram negative, it is made up of 3 layers. In gram positive, it contains inner cell membrane and outer cell wall, and outer only cell wall, which is made up of peptidoglycan layer. Whereas in case of gram negative, it contains 3 layers: one inner cell membrane and outer cell wall, middle cell wall, and there is an outer membrane called lipopolysaccharide layer. Here you can see the gram positive. You can notice cell membrane here, and after to that complete peptidoglycan layer in case of gram positive. Whereas in gram negative, you will notice inner cell membrane, and in middle there is periplasmic space that is common to the both, and after that there is a peptidoglycan layer where it is thin peptidoglycan layer in case of gram negative, whereas in gram positive it is very thick. And in case of gram negative, after peptidoglycan layer there is one more layer called, that is the outer membrane made up of lipopolysaccharide, the outer, which is top to the cell. So here we will find three layers in these two layers. Function of cell bar, it provides a rigid structure to the cell, and most are rigid because of peptidoglycans content. So gram positive cell, you can see sheath of peptidoglycan, as I told. So these are the structure of gram positive and gram negative. So peptidoglycan is nothing, I also called as murine layer, and they are made up of, peptidoglycan is made up of two alternating uh, sugar sugar polymers, that is beta 1,4 N acetyl glucosamine and N acetyl muramic acid. So these are alternatively arranged in this peptidoglycan layer. These are the peptidoglycan is made up of these two molecules. So next, coming to the L forms. L forms are nothing but uh, L forms arise naturally from mutation in the wall forming gene, and but they are also can be induced artificially by the treatment with some enzymes which disrupt the cell wall. Here, if the gram positive cells are treated with some enzymes which dissolve the peptidoglycan layer, then it becomes a protoplast. Whereas with the gram negative cells, mm, as it contains outer membrane, that is lipopolysaccharide, it doesn't completely remove the external layer, and this forms spiroblast in case of gram negative. So here you can see the gram positive, when peptidoglycan is lost, it forms protoplast, whereas in gram negative, when peptidoglycan is lost, it forms spiroblast. So it is a cell membrane. We also know contains phospholipids, proteins. There are some exceptions like mycoplasmas and archaea, and mostly these are selectively permeable layer, as we all know. It allows only certain kinds of material, and also used for secretion and energy reactions.